All right, the following set of notes is our last phase of cellular respiration, which is the electron transport chain. So as I mentioned previously, if you haven't done this already, you want to make sure you watch this video. And this video can be found on the course Moodle, right? So just under our regular videos, under cellular metabolism, if you click on the videos, and just down at the bottom here, page 6, overview of electron transport chain. So this gives you a good visual first of what's going on, and then we'll kind of go through the main points of what's happening during the electron transport chain. So if you haven't watched this yet, pause these set of notes, go and watch this video, and then come back to these notes. All right. So just to kind of recap some of the things that we've talked about in the first two stages, if you recall, the primary purpose of the first two stages was these series of oxidation reactions in which there was a loss of electrons, right? So glucose being the primary source of energy to start the reactions is giving up electrons to these high energy electron carrying molecules called NADH and FADH2. Now, we should make sure we understand that NADH comes from, we get a couple molecules of NADH from glycolysis, and then during the Krebs cycle, we get about 6 molecules of NADH, and we get a couple of FADH2. Okay, so just keep in mind that these are our electron-carrying molecules that now enter into the final stage of the electron transport chain. Okay, so we got NADH and FADH2 are the high energy electron carrying molecules which are going to provide the energy for the bulk of ATP production in cellular respiration. So remember at this point, glycolysis and the Krebs cycle have each yield about 2 ATP, right? And yet from one glucose molecule, we can get upwards of 30 to 38. So that's kind of leaving us in the range of sort of 30 or maybe 28 to 34 ATP are going to come out of the electron transport chain. So if we look at the diagram down below, we'll go through and just kind of see what's happening from a visual, and then we'll kind of summarize it in the notes down below. But if you remember, this is kind of the picture we've been using to represent the molecule NADH. We'll kind of use the same thing for FADH2. The only difference really between the two is where they pass their electrons into the electron transport chain. So what we want to remember in this is this phospholipid bilayer is the comprising the inner membrane of the mitochondria, right? So that's the inner membrane of the mitochondria. And now we get another good look at the mosaic model of that membrane and how we can see these embedded proteins are going to play a significant role in the series of chemical reactions that take place. So we got NADH is going to pass its electrons on to this first integral protein here, and FADH2 tends to pass it along to this protein that's embedded, which is more of a peripheral protein. This protein, much like we talked about before, remember the cell membrane is dynamic, right? And that means that there's fluidity to it, right? And these proteins aren't static, but they can in fact move along. And all of these proteins have been identified and have specific names, but we don't need to worry about that right now. So we're just looking at the fact that these high energy electron carrying molecules are going to pass their electrons along to these proteins embedded in the cell membrane. And as they do so, of course, then they go back to their original forms, NAD+, right? As it gives up 2 electrons, it will return to NAD+, and FADH2 will return to FADH, right, as they give up these electrons. Now, in this process, so again, we've got this series of redox reactions, essentially, right? So as one substance, the NADH or FADH2, lose electrons, right? We've got an oxidation, and of course, as the proteins accept electrons, they're gaining electrons, then they're becoming reduced. So again, it's this coupling of an oxidation to a reduction reaction. And with that, energy is released, and that energy released is used to move these protons, these hydrogen ions, across the membrane. And so what we're doing is creating, essentially, we call it a proton gradient. And these are all hydrogen ions. And the reason why we can refer to it as a proton gradient is, if you remember, the atomic structure of hydrogen has just one electron. So if we remove that one electron and create a hydrogen ion, that hydrogen ion has a +1 charge because it's simply a proton. So we often refer to this as the proton gradient, even though it's a high concentration of H+ ions. So all along the electron transport chain, electrons are being shuttled from one protein to another, and that energy released from there, pumps the protons from the inner membrane across to the outer membrane. So we've got a high concentration of H+ ions on the outside. All right. And the electrons continue down their path from one enzyme to another along the inner membrane. And this high concentration of hydrogen ions now provides the energy here at the end to drive ATP synthase, right? So we've got the flow of hydrogen ions through the enzyme ATP synthase, releases energy to drive the endergonic reaction, right? The endergonic reaction of basically the phosphorylation of ADP. So we're going to add this phosphate onto our ADP, creating ATP, which is occurring right here, right? And that's the enzyme ATP synthase. So we get our ADP plus PI, inorganic phosphate, converted into ATP. And of course, as mentioned earlier before, right, this little red molecule here is our oxygen, and the oxygen becomes the final electron acceptor in the electron transport chain. And as it accepts the electrons, it also accepts along with it a couple of hydrogen ions, and that forms our water. And that equation, I think we'll write it down below, but just to let you see it a few times, that equation looks like this: an oxygen molecule plus 2 electrons plus 2 hydrogen ions forms our water molecule. One thing in terms of, we talked about before how important it is that chemical equations are balanced. In reality, we say, well, we only need half of the oxygen molecule to form one water, right? Because the oxygen molecule is diatomic, meaning, there's 2 oxygen atoms there. So we're going to use one of those oxygen atoms that's going to gain 2 electrons and 2 hydrogens to form our water. So that's kind of the technically balanced form of that equation. All right, so that's the bulk of how the ATP is synthased. is how the ATP is synthesized. And even though the oxygen is appearing here at the end of the process, it's still a reactant because it's accepting electrons forming water. So if we go back to our original chemical equation of cellular respiration, we've got C6H12O6, which is our glucose, and we say that it reacts with oxygen, which is really kind of funny to say that, right? In the sense that they really don't directly react together, but the glucose molecule has passed its electrons to these intermediate molecules, which then shuttle the electrons eventually to oxygen, but indirectly, right? It's not from directly from glucose to oxygen. So we got glucose reacts with 6 molecules of oxygen, producing 6 molecules of CO2, 6 molecules of water, right? That's where we see our water, and then our molecules of ATP. And again, just like nothing is perfect, depending on the number of protons that have been pumped across, right? That's why our number of ATP may vary anywhere from, 30 to 38 ATP in a per given 1 molecule of glucose. So down below here, we summarize these. The final stage involving electron transport chain occurs within the inner membrane. The electron carrying molecules, I'll just switch to my highlighter here, but the electron carrying molecules NADH and FADH2 are now losing their electrons to several protein intermediates. And by protein intermediates, these are essentially enzymes, right, that are catalyzing, again, different steps of the metabolic pathway, right? And so those electrons are passed to those proteins, those enzymes, embedded within the inner membrane of the mitochondria. So this oxidation or loss of electrons creates the energy that drives the transport of H plus ions across the membrane from low to high concentration. So here's that transport. It's active because it's requiring energy, but interestingly enough, in this case, the energy doesn't come from ATP, but rather the exchange of electrons between molecules. And we get this electrochemical gradient, which is similar to the chemical environment of a battery, right? So when you think about how a battery powers your various devices, the cell itself is acting like a battery by creating this distribution of charge with hydrogen ions on the outside, creating that potential energy to drive the formation of ATP. So the diffusion of H plus ions through the final integral protein, and that final integral protein being called ATP synthase. We go back to the naming, right? Enzymes, they end in the ASE, ACE. So this ATP synthase creates the energy that drives the synthesis of ATP through a process called oxidative phosphorylation. And this is an important distinction here because the energy driving the process is coming from that loss of electrons. And it's important to distinguish that from back in glycolysis. If we go back to our notes in glycolysis, where we talked about enzyme substrate phosphorylation. So remember what was causing the phosphate to be added to ATP in the formation of ATP in glycolysis, right? This was driven by an enzyme, whereas in the electron transport chain, the phosphorylation of ATP is driven by chemiosmosis, the proton gradient, which was established from the oxidation process, the transfer of electrons. So one is referred to as oxidative phosphorylation versus in glycolysis, it's referred to as enzyme substrate phosphorylation. And finally, the molecule in red, oxygen, right, that accepts the final electrons reemphasize that equation in which the oxygen is going to accept 2 electrons along with 2 hydrogen ions in the formation of water, right? And we should really recognize, much like in the Krebs cycle, right, and going back to our equation of cellular respiration, that we get these molecules of CO2 and water. The CO2, if you remember, was generated during the Krebs cycle, and the water generated during electron transport. These really are waste materials that are removed, right? So as they build up inside of cells, carbon dioxide and water builds up in cells, those will move via osmosis or diffusion out of cells into the blood, and either in the case of CO2 and water, they'll be brought back to our lungs and we'll breathe them out. And also our kidneys play a role in maintaining the water balance and water levels in our blood in order to maintain blood volume and blood pressure. So kidneys may play a role with water as well as the lungs, but just the lungs are dealing with the waste of CO2. And that's it. See you Monday.